species of benthic foraminifera which thrive very well because they are adapted to low oxygen environment. Another advantage is that usually we find very high population of benthic foraminifera in oxygen minimum zone. Then the question comes up, if there is low oxygen, why the foraminiferal population is increasing? It's because the higher organisms which feed upon those benthic foraminifera are not there. Or so to say, the predatory pressure is less. The larger organisms, they eat those sediments and they destroy the foraminiferal shells. Since the dissolved oxygen concentration goes down very low, those bigger organisms are unable to survive. They are less in number, and that's why they don't feed upon the sediments, so the benthic population increases.
Okay, so now there is one question from Miss Madhurima Ganguly. How is the ratio between planktic and benthic forams maintained for understanding the sea level changes owing to the dissolution of calcareous dust below the CCD?
See, if you start moving from a beach towards the shelf and deeper region, you will find more of benthic foraminifera on the shelf than planktic, because planktic foraminifera, they need a particular depth of the water column for reproduction as well as for their living. So initially, we don't get planktic foraminifera. Planktic foraminifera, we start getting from say 60, 70 meter onwards. So try to imagine there is almost 100% benthic foraminifera. That ratio changes as we go into deeper water. Slowly, you will get say 90% benthic foraminifera, 10% planktic foraminifera, then 80% benthic, 20 planktic, and then at some point 50% benthic, 50% planktic, and then planktic foraminifera will increase, then the benthic foraminifera. So what do we get? That if with increasing water depth, the ratio of benthic to planktic foraminifera decreases. At shallower depth, benthic foraminifera are more, and at deeper depth, planktic foraminifera are more. And that's how we use this proxy to understand these sea level changes.
Okay, so to all the participants, we are having some trouble with the YouTube live. So we think we will be just extending our questionnaire session so that the technical team will be able to go to the YouTube live once again. So to Humne sir, we will be waiting 5 more minutes. By the meantime, we will be having the interactive session with the Dr. Raji Sarso sir. Okay. So sir, there is hope you are fine. So sir, there is one question from Mr. Rizwan Khalil. How can we determine the paleoclimate condition by the determining the isotopic composition?
See, that requires a too detailed an answer and another lecture on that. Anyway, I'll try to summarize that very quickly. See, the seawater has different isotopes of oxygen. It has lighter O16 and heavier O18. When evaporation takes place, it preferentially removes the lighter isotopes out of the seawater. So the resultant seawater is enriched in the heavier isotope of oxygen, that is O18. Now, foraminifera secrete their calcium carbonate from that seawater. And foraminifera also have this calcium, carbon, and oxygen. So they take up whatever available or the ambient isotopic composition is. So if there is more warmer condition, although there will be more evaporation, but then at the same time there is melting of the ice sheets. Ice sheets have comparatively lower oxygen isotopic composition. So that fresh water that comes from the ice sheets, that increases the lighter isotopic composition of seawater. And that's what the foraminifera incorporate. That's one way that we use the foraminiferal isotopes for temperature condition or the freshwater influx also, and directly for temperature also it can be used. Arun, you are not audible, sorry.
Sorry, sir. My mistake. So there is one more last question from Ms. Ragini Yeshwanti. So if you are getting foraminifera on the land surface and that too in a marine surface, then what are the pros of the macerations? Is there any difference between the maceration techniques?
Yeah, the way I told that freeze drying, definitely it is not going to work, because there you have to make small pieces of those consolidated sediments, because it will be difficult to freeze dry and then dissociate them. So the processing for those consolidated sediment or the exposures on the land is different then what I told it, this processing works basically for the recent sediments or the marine sediments.
Okay, sir. Now there is no more questions, sir. So thank you, sir. Now I would like to welcome Dr. Devi K, Assistant Professor, Department of Geology, for proposing the auto thanks to our speaker. Devi, miss, over to you.
Tarun, just a
Yes,
minute.
yes, sir.
I think Professor Gandhi has asked a question. Can I answer that? I see that in the chat box.
Sure, sir. I didn't see the question.
Professor
Sorry.
Gandhi asked, is there any standard procedure for subsampling the cores samples 2 centimeter or 5 centimeter? Professor Gandhi, I would request that please subsample the core at 1 centimeter. And afterwards, you can use every second centimeter or every fifth centimeter. So if you have a core, please, it will take little more time, but please subsample at 1 centimeter intervals and afterwards decide whether you want to work at every 5 centimeter or 10 centimeter.
Okay, sir. Thank you, sir.
Okay, sir. There is, sir, Rajiv, sir, there is one more question from Dr. Vishwanath. How to avail these sampling and analytical facilities? Are these facilities available only on NIO?
Yeah, see, I can understand ship definitely is only with NIO. You can't have, or the institutes cannot have the ship. You can write to, say, Geological Survey of India or, say, NIOT. They can provide you ship. But yes, multi-corer as of now, or to the best of my knowledge, that's also available with us. Only no other institute in India has the multi-corer. But you can always have a freeze dryer or a freezer. I think these are basic facilities that you can get in any lab, or if not your lab, maybe chemistry lab or a biology lab will have a freezer or a lyophilizer. You can, or if not, then I mean, it doesn't cost much. The entire system of freeze drying that is comprising of a freezer and a freeze dryer going to cost you maybe around 6-7 lakhs. So if you can write a small project or if you can get small grant from somewhere, it's easy to have this facility.
Okay, so since we have got our tight time schedule, since we are late, so can I share your mail ID with the participants? So that
yes, definitely. Please do that.
Okay, sir. I will be sharing your mail ID. So if they have any doubts or any query, they will be contacting you, sir. Thank you, sir. Thank you for the session, sir. Now I would like to invite Dr. Thank you, sir. I would like to invite Dr. Devi K, Assistant Professor from Christ College, Irinjalakuda, to propose the formal auto thanks to the speaker. Devi, miss.
Thank you, Tarun, sir. I would like to thank Dr. Rajiv Saraswat for this informative talk of recent developments in collection, storage, and processing of sediments for foraminiferal studies. Thank you, sir.
Thank you. Thanks a lot.
Thank you, sir. Thank you, sir. Have a nice day, sir.
I'll stop. I think I already stopped sharing my screen.
Yes, sir. You have already stopped sharing, sir.
Yes. Yeah. Thanks.
Okay, thank you.
Our next speaker is
Devi, miss, you can invite the next guest.
Our next speaker
is you can continue.
Dr. Sumit Humani, Professor, Postgraduate Department of Geology, Rashtrasant Tughadoji Maharaj, Nagpur University. He has been 19 years of teaching experience. His field of research include applied and industrial micropaleontology, quaternary specializations of geology and limnogeology, paleoclimates, paleolimnology. He has membership in various professional bodies like IPA, IAFA, IOP, and a lot more. He is a joint editor of Journal of Geoscience Research of Gondwana Geological Society, Nagpur, coordinator of Nagpur Student Chapter of Paleontological Society of India, Lucknow. He has completed research projects of SERB, UGC, DST, FIST. He has visited Süleyman Demirel University, Isparta, Turkey, Climate Change Institute, University of Maine, USA, and he has published more than 60 research papers. He has also been a reviewer of more than 10 national and international journals. I welcome you, sir, in this webinar. Over to you, sir.
Tarun, am I audible?
Yes, sir. Yes, sir. You are audible, sir. Over to you, sir.
Okay, thank you very much. Very good afternoon to all of you. It's almost 12 o'clock. Half an hour we are delayed. So anyway, but we'll start with the presentation. I'm going to talk on the diatoms. So recently, Saraswat spoke on foraminiferal applications and the techniques to sample the forams. So now I will be taking you to the journey of diatoms, applications of diatoms in applied geosciences. Myself, Dr. Sumit Humain from Applied Department of Geology, Nagpur University, Nagpur. Now coming to what are diatoms. So diatoms are unicellular microscopic single cell algae predominantly made up of micro siliceous algae, siliceous composition. It has a well developed, beautiful, complex cell wall of silica. You can see in the picture. They are the major photosynthetic producers on the earth. The diatom needs sunlight to generate their own food. So therefore, majority of the diatoms you will find in the aquatic environment. So they will be dwelling at a shallow water depth of the lakes, in the oceans. They are ubiquitous. They are found in almost all aquatic environments. So right from freshwater to brackish water and in the marine realm as well. So they are the major, they are playing major role in the global carbon and silica cycles in the oceans. These diatoms, they are having different modes of life. So these diatoms, they live in the water column. They are called as planktonic diatoms. So other diatoms, they dwell at the bottom of the oceans or the bottom of the lakes. So these diatoms vary in the locations at the depth of the water bodies. So it could be epilithic. They attach themselves at the bottom of the rock, and they are referred as epilithic diatoms. Some diatoms, they attach themselves to the sand and live at the bottom. They are called as episamic. Some diatoms, they live at the in the mud at the bottom of the water bodies. They are epipelic. Likewise, some diatoms attach themselves to the planktons which are present, plants, they are present in the water. They are called as epiphytic. Likewise, their benthic habitat varies. Then other diatoms which are floating in the water column, you may call them as a planktonic diatoms. See, these diatoms are very much sensitive to environmental change. So therefore, they are the obvious choice of the major geoscientists to study the organisms to solve the geological problems. They are sensitive to nutrient changes. They are sensitive to physicochemical changes in the water body, particularly pH, conductivity. And interestingly, they are made up of, since they are made up of silica, they have got the well-preserved crystals or the cell walls in almost all sedimentary environment. You are going to get the diatoms present in the almost all sediments, right from marine to freshwater. Now, how to identify these diatoms? So you see these pictures. Diatoms, they are made up of basically, so they are the single cell, but they are made up of two valves. One valve is called as epivalve, which is lying above. You can see this one valve of the diatom, whereas another valve, which is lying below, this is called a hypovalve. So likewise, the diatom is constituted of, or they are made up of two valves. In between, you have the different bands. So these bands are called as girdles or the cingulum. So with this diatom is, in this way, the diatom is made up of. So these diatoms, basically, they are commonly found in two shapes. You can see the circular pattern of the diatoms. So they are called as centric diatoms, whereas another type is there, that is the elliptical or elongated one. It is the pennate one. So you can see these diatoms has got the epivalve and the hypovalve, epi and the hypovalve, and in between you have the cingulum. Why it is essential to understand this morphology, because once you want to apply, because these diatoms can be identified up to specific level with detailed variations. So therefore, we need to understand first why, how these diatoms to be identified. You can see the diatom has got the fine perforations or the pore present here. These fine perforations, when they are arranged in a line, we can refer them or we can call them as a stride. You can see the stride, and in the center of the valve, you have the median line or the slit. So this is called as raphe. Raphe may be extended from poles to center area of the diaphragm valve. So this raphe and the striae, they are playing very vital role in the identification of the diaphragm, maybe the centric one or pinnate diaphragm. So to identify the diatoms, in addition to identifying the centric pinnate, we need to identify the different shapes. So diatom has got the various shapes like linear, ovate, circular, lunate. So these are the basic few shapes I have given here, so that you will have the idea how to identify the different shapes with the different names. So you can have the sigmoid shape diatoms as well. So in between the two valves, just now I showed you that in between the two valves, EP and the hypo, you have the girdle or the cingulum. You can see the girdle view also varies from species to species. So few examples I have given here. It could be the rectangular one, it could be the clavate one. There are many other examples of girdles. So apart from the girdle, you need to take into account the apex, how the apices of these diatom look like. You can see this diatom has got the obtuse apex, whereas another one is capitate, third one is the sigmoid. Likewise, you need to identify the apex pattern also. Most of you must not be knowing how to identify the diatoms. So for them, most of the students are here in the workshop. So for the benefit of them, I have included these figures, how to identify these diatoms. So based upon these characters, you can broadly categorize the diatoms into two. One is the centric and another is the pinnate. So centric diatoms, basically they are circular in pattern. I told you it, and just now I showed the figure also. And the pinnate ones, they are most diversified groups. They are again divided into arapid, monorapid, prorapid, and birapid diatoms. So now, when you talk about the raphe, the diatom valve, when you have the raphe present in both the valves of the diatom, it is called as birapid diatom. Whereas sometimes in some varieties you have the raphe in a single valve. It is, I'm going to show you the figure also. It is called as a monorapid diatom. Whereas some species, they don't have the raphe at all. They are also referred as arapid diatoms. Whereas some diatom species, they have very short raphe present at the poles. So that is called a prorapid diatom. So you should identify the raphe pattern also to know the species. So with these, you have the birapid diatoms. They are again categorized into naviculoids, sigmoids, clavates, emboloids, raphe, keel. Likewise, details I will skip these. Now, in the diatoms, for your information, quickly we'll discuss this. The diatoms has got the peculiar type of cell division. So diatoms, I told you that they are made up of two valves, like epi and the hypo, like a pill box. One valve, it is epi, lying above, and lower one is called as the hypo. So just hypo wall is little bit smaller as compared to epi wall. So they get fixed into each other. And it constitutes the diatom. So the first generation, when the daughter diatom produced, it is again having the same size, but in the second generation, what happens? The hypo wall again divides into EP, and the next third generation EP and hypo wall. So now the size of the diatom goes on reducing. In this way, there is a continuous decrease in the size up to certain level, and again the sexual reproduction takes place to increase the size. Likewise, this diatom reproduce in the water bodies. You can see the details of SEM image of the diatom, how they look like. You have the slit extending from the poles to the center, and here you have the area which is not ornamented, plain area. Again, the wall extended here, and you have the hook-like structure at the poles. Likewise, these characters are very important to identify these diatoms. Now, these fine perforations of the pores, you can see very clearly here. They are arranged in a line. So therefore, we call them as a striae. These pores are referred as striae. The diatoms, again, you see that the diatoms could survive in the extreme environment as well. For example, the extreme drought conditions, no? So the diatoms develop the oxospore. So under the unfavorable conditions, when there is no rains or drought, so they form the oxospore and they keep idle there for waiting for the favorable condition to develop. Once the favorable condition develops, the diatom starts growing again. So in this way, the diatom species could survive in the extreme conditions as well. So this is very interesting phenomena found in the diatoms. Now we'll talk about how these diatoms look like. You can see the centric diatoms. Centric diatoms, mostly they are circular, but if you see this species, Zoloposita, here you can see that it is present in the girdle view. Majority of the time it is present in the girdle view. If you see the valvar view, you can see its circular pattern. So it has got the striae arranged in a line, or it may be zigzag in nature as well. This this cyclotail, another one is very clear circular pattern of the diatoms. Some diatoms, centric diatoms, they have the eye-like structure. Can you see here? These 4 dots are there. The eye-like structure may be the typical identifying feature of this one of the centric diatoms, Pantoxaceae. Here you may get the 3 eyes, 4 eyes, or 5 eyes. Likewise, you should use these characters to identify the centric diatoms. Now coming to the pinnate diatoms, pinnate again it is the most diversified group, or elongated diatoms. You have the araphid diatom. I told you in the beginning when we discussed the classification. Here you can see this Alneria. Earlier it was called as Senedra alna. Now recent nomenclature has changed the name to the Alneria, so we should use the Alneria. Here you won't find the raphe. Here you won't find the raphe in the another sample, Fragillaria. So these type of diatoms are called araphid. In both the valves, you won't get the raphe. In the monoraphid, you have these. You can see this example here. Coconis here, you have raphe. Here another valve, you don't have the raphe. So therefore, it is monoraphid. Some diatoms they are prorapid. I told you the raphe is seen along the poles only. That too, you can see it in the girdle view. This photograph F, Eunocia. Here you can see the girdle view. So in the raphe here, Eunocia, raphe may not be seen. Now third type in the in pinnate is the birapid naviculate. You have the raphe in the both the walls. Example, Diplonis, Straticula, Navicula. I'll quickly go through this. Birapid sigmoid, sigma-shaped structure very clearly. You have the birapid clavets. So clavet is the tapering form. From one end it is tapered. You can very clearly identify. These lines are in the light microscope. You can see the pores as a line. But when you study it under the scanning electron microscope, this line would look like a pores. So when these pores are arranged in a line, these are the striae. And this is the example of clavet, that is the Gomponema. You have the birapid symbioid types like Amphora, Symbella, and Ansoenema. How to identify the Symbella and Ansoenema was just one example. You have to concentrate on the raphe. This raphe is turning towards the dorsal side in case of Symbella. You can see this photograph. So this raphe is turning towards the dorsal side. Here, in case of Ansoenema, the raphe is turning towards the ventral side. Likewise, you can use these features to identify these diatoms. You have the birapid diatom with raphe on kill. Here you have the kill elevated portion, or the ridge of the silica. Here you have the raphe here somewhere. You have the transapical costi here in case of denticula, epithemia, Rhopalidea. Costi is nothing but the unornamented or unperforated ridge present between the striae, that is costi. So you have the birapid diatoms with the strong kill margin or the strong fibulae. You can see this fibulae, it is nothing but the breach of a strong siliceous structural breach. It is made up of silica, which is lying over on the canal of this raphe. Here raphe is there on the sides. Here you have the horizontally developed fibulae. So these fibulae are made up of the bridge-like structures made up of silica, and this is the typical characteristic feature of this genus, like Hansia, Nicia, Triblonia. You have the raphe encircling or circumference around the wall. You have the example is Surirella. Here you can see the raphe is all along the wall of the diatom. Now, with the advent of the technology nowadays, a lot of nomenclatural changes are there. So there is urgent need to study these diatoms using scanning electron microscope. In the light microscope, you cannot see some of the features that could be seen very clearly in the scanning electron microscope. Therefore, I have shown you this figure. You can see the Cyclotella meningraniana. On topmost figures, you can see the light microscope images, whereas in the scanning electron microscope, you have the detailed characteristic features. You have the Fultopoltuli FB. These are the tube-like structures present above the costi, and in between the two costi, you have the striae. Here you can see the striae. Here you have the costi, and above that, the tube-like structures. You have the Fultopoltula. And in between, you have the vertical pores, or you may say sometimes leaf-like structures. So labiate structures, they are also referred as remoportal. So in this way, there is a lot of variation when you see the light microscope images and the ACM images. So this is an intertrapian diatom from central India. Now, paleolimnology, coming to the paleolimnological approach, just now Dr. Saraswat spoke on how to collect the samples, but in the lakes also a lot of difficulties are there in collecting the samples. So we generally go to the lake to collect the samples using the gravity corer. Here we have collected the sample using the gravity corer. The topmost, we need to take into account that the topmost sediment sample must not be disturbed. Once you collect the sediment samples, you need to process it in the laboratory. You have to date the samples by the lead 210, C14. These are some of the examples of how the sediment dating is done. Then you should process these diatoms for the samples for the diatom studies, and identify, enumerate under the microscope. This is in general we study. So topmost portion is representing the recent samples, and we need to correlate it with the water quality parameters, physicochemical parameters. And then you have to study the large number of data sets. You have to collect the data set from the different environmental conditions, having different pH, salinity, temperature ranges, so that the diatoms which are sensitive to those environmental conditions can be calibrated in a data set. And using the transfer function, you can derive the models and diatom diagrams, interpreting the paleoclimate, paleo-rainfall, or paleoecology of a particular area. Now, quickly, we should discuss how to collect how to process those, macerate the diatom samples. So, the standard method is given by the Butterby. So, once you collect the sediment sample, you need to process it with the SCL, depending upon the geology present in your study area. If the carbonate rocks are there, then you need to process it with the SCL. But whenever you collect the sample, use the dilute SCL to process the sample to dissolve the carbonates from the sample. If the carbonates are not there, you can directly switch over to the H2O2. 30% H2O2 is generally used to dissolve the organic matter from the sample. You need, if the size of the sample organic material is larger, then you can screen it. If it is attached to the clay material, you can use the dilute ammonia also to release it from the clay fractions. And you can prepare the test slide. You can count you can see the diatoms in the microscope. If the sufficient number of samples are there, then you can prepare these slides and you can do the enumeration. If sometimes people also use the microsphere to count the diatoms. Now, coming to the water quality and the eutrophication, without understanding of pre-settlement water quality conditions, practical management for lake water quality are very difficult to establish. So therefore, existing water quality of the data of the lakes or any water body, if you see, it doesn't extend to the pre-settlement period. Therefore, it is not known to what extent settlement, sewage disposal, and other changes in the lake basin specifically have changed lake water quality. So, therefore, the diatoms are used as an indicator to know the past water quality and climate because the diatoms are important component of the ecosystem. They are widely distributed in many habitats. Their siliceous remains are helping them to preserve nicely in the almost all type of sediments. You can identify these diatoms up to lowest level, that species level. Individually, you can very clearly identify. The diatoms are always present in large number in most of the samples. So they have got the strong correlation with the environmental characteristics. Even if there is a change in the 0.1 pH, you will see there is a change in the diatom pattern. Such a sensitivity is there for the some of the diatoms. So they are showing the rapid change to the responding to the rapid change in the climatic conditions. You can see they have the efficient storage of representative samples. So when you study the diatoms, you need to correlate it with the water quality, topmost samples of the core. You have to study, compare it with the physicochemical parameters like pH, salinity. So you can use the two models. One is called as the linear model. So here, the linear relation between abundance value, you can see the abundance of the diatoms here with the and the environmental variables like pH, temperature, salinity. You can compare and you can study the linear model. You have the another model to study the diatom, that is the uni model. Here, abundance value of the species, you have the abundance value with the environmental parameter. You can compare likewise. The diatom correlation is done with the water quality parameters. So eutrophication trends can also be studied. This core is representing, it is from Liu et al. 2006. So you can see how the core is prepared to indicate the total phosphorus concentration. And it has been correlated with the directly measured data also in situ sample. So that whatever you have interpreted it, based on the diatoms, whether it is matching or whether it is correlatable or not, that can be confirmed. Diatoms are very good indicators of salinity. Some species have got the narrow range of salinities. You can call them as a stenohaline. Without such restrictions on the diatoms, you can consider them as cosmopolitan. Chromatophora is one of the examples. You can found in the brackish water, marine environment. You won't get it in freshwater environment. You have the freshwater diatoms. In that, there are some species which has got the little salt tolerance. You can call them as a halophilic, occurs in the coastal lakes. Whereas the freshwater species, you can consider them as a stenohaline. These species cannot survive in the saline environment. There are diatoms which are very good indicators of paleo pH. I told you, even if there is a change in the 0.1 pH, some of the diatoms may have the sensitive to these changes, and you will have the variation in their pattern. So diatoms which are having pH less than 7, these diatoms called as acid-living diatoms. You may call them acidobiontic diatoms. Some diatoms, they are found at circumneutral pH around 7. Some diatoms, they are alkylobiontic, so more than 7 pH. So likewise, you can categorize them. So the pH concentration favorable for the healthy fish population, what is observed is 6.5 to 8.5. So that can be determined using the diatoms also. These indicators of diatoms, they are very good indicators of trophic status. You can count the total number of diatoms. More the diversity of this diatom, more the productive water body. Likewise, this is a very important parameter to identify the productivity of the water body. Centric to pennate ratio, circular to elliptical ratio, you can count. The more the centric, more the productive environment. Because the centric diatoms, they are always found in the water column. So they are the first to be affected with the nutrient change. So centric to pennate diatom is very important. indicator. There are some indicator species as well. Likewise, Trypanodiscus is one of the important indicator diatom, which is always found in the eutrophic water. You have the oligotrophic water bodies also. So these are being represented by the genus like Tabellaria. They are always found in the oligotrophic environment, wherein the nutrient concentration is very low. The planktic to non-planktic ratio is important. Planktonic forms are more common in eutrophic water, considering the abundance of the nutrient in the water. So diversity indicators, less the diverse, right, the stressful will be the conditions. So there are two extreme conditions. One is extremely trophic, highest trophic status, that is the hypereutrophic or hyperoligotrophic. Both are the stressful conditions, and they indicate the some kind of pollution in the water. So the items can also be used to study the lake level fluctuations. There exists a strong correlation between diatoms and ionic concentrations. So you can reconstruct the past changes in the lake water salinity given by the hydrological and climatic changes. So, for example, you can identify the species like Rupulidea. This is a very good indicator of the diatoms found in the drier areas where you have the higher salinity compared. You can get the abundance of this benthic diatom. Whereas sometimes you get the dominance of Holocosida granulata. It is most commonly found in the eutrophic water bodies. Sometimes it is there in the mesotrophic water body as well, but mostly it is found in the eutrophic water bodies. You can use these diatoms for the stable isotope studies, oxygen isotope studies, to understand the variation in the paleo temperature of the water. The diatoms, they have very good role in the industry also. They have the economical importance. The freshwater diatomites rarely exceed in 1 centimeter. The diatomites, these are the particularly sedimentary rocks formed by the diatom deposition. So this could be possible only in a situation where the rate of deposition of diatom crystal is higher than the other sediment components. For example, in our tropical areas, we won't expect the development of diatomites. We could get the abundance of the diatomites where in the cooler conditions where the other sediments are not being supplied in the higher concentration as compared to the diatoms. Only the diatoms will form the basis of the sediments. Then only you are going to get these diatomites. So diatomites mostly you will get it in the European and colder areas in the world. So they are also very useful in the filtering, sugar industry, refining, toothpaste industry, abrasive, polish, paints, lightweight bricks. Likewise, there are many various industrial applications of the diatoms. So the diatoms, they have got the uses in the biostratigraphy, in the petroleum exploration, mostly the Cenozoic diatoms. The diatoms, they are generally recorded from Mesozoic onwards. So their use is commonly found in the Cenozoic diatoms, mostly used in the petroleum exploration. You have the other applications of the diatoms in the marine oceanography, coastal and estuarine studies, archaeology and forensic science. You have their applications in nanotechnology due to the structural designing of the diatom. So it is being used by the to prepare the architectural design in the different parts of the world. So now coming to the lake sediment, how we study. So the lake sediments are the very good repositories of natural these sediments. So you can have the terrestrial climatic variations by studying the lake deposit. You can understand the paleo temperature, precipitation patterns. You have the annual banding. You can see in this diagram. This is from our central Indian lake. You can see the annual well-developed banding. This is caused by the seasonal changes in the productivity and the variation in the input of the sediments. So mostly in the summer, you have the biologically rich and organically rich deposit, whereas in the monsoon, you have mostly the runoff deposit. You can very clearly see here. You can analyze them for isotope studies. You can retrieve the diatoms and osteochord. There are different methods. I am not going to give here, but there are different methods to study the stable isotopes from the diatom crystal or from the osteochord shell. You can see here another core. Here you have the lighter bands and the darker bands are very clearly distinct. They are the indicator of variation in the climatic conditions, wet and dry conditions. You can interpret based upon the size of the diatom. Mean linear diameter is very important factor that you should analyze. Mean linear diameter of the diatoms. So smaller size diatoms, they are implying to the drier conditions, abundance of the smaller size diatoms. Whereas the abundance of the larger ones, along with all types of diatoms, this larger to smaller one, they are commonly found in the wetter condition, comparatively cooler condition, because of particularly in the central or tropical region I am talking, wherein you have the abundance of the diatoms because of the availability of the more nutrients during the monsoonal condition. During the dry periods, there is a less availability of the nutrients, so diatoms do not grow to the large size. Their population will also be restricted, so their size will be minimum. So smaller size diatoms, they are indicating the drier conditions, and the larger size diatoms, they are indicating wetter conditions. Likewise, we studied the several lakes from central India. So I told you that we should correlate the topmost sediment sample, the diatoms from the topmost sediment core with the water quality parameters. So you have the, for example, the Alocosira granulata, you have the pH of 8.1 to 8.5 something, which range is there. Once you identify the relationship of the surface water quality with the diatom, you can use them to extrapolate it in the core samples. So that is very important. Unless you have the present day database, you will not be able to talk about the past. So present day database generation is of utmost importance. So you should first develop that database, and then only you can use, because Indian diatom database is unfortunately is lacking. We have the American diatom database, we have the European diatom database, but we don't have the Indian diatom database that can be used to interpret the past water quality status or the past climatic status. So here you can see how sometimes some diatoms are common in the almost all conditions, so you need to take into account the other type. For example, here you have the dominance of Alocosira granulata in almost entire core. You should see this another type, Cyclotella meningiana, is found in the mesotrophic eutrophic environment in response to this benthic diatom in the same. Another core is there. We have the pH changing. Another diatom patterns are also varying. You can correlate it with the stable isotopes and total oxygen nitrogen concentration ratio also. TOC, the total nitrogen ratio, is compared with the stable carbon isotope. You can identify the origin of the organic matter in the water bodies. Van Travelin diagram will tell you how the hydrogen index and the oxygen index is used to identify the origin of organic matter. So likewise, different lakes we have studied here. You have the another lake in central India as well. Pre-monsoon, post-monsoon pH level, electrical conductivity also varying. So in response to that, the diatom speciations are also changing. So this is very interesting. You need to use it to understand the past qualities. So in this diagram, you can see that the past water quality or the past rainfall correlation is also correlated within the diatom size. This data is available in the meteorological department. You can download it and you can correlate it with the size variation. Likewise, other lakes. So here also different regions we have tried to understand. Now very soon we are going to produce one database for the central Indian region, the diatoms and the water quality relationship. So diatom succession is you can subsequently interpret using the different level of correlations, mainly using the conies, that is the constrained incremental sum of squares. So using the Telia software, you can do this conies, so that you will be able to identify the relationship of the diatoms in the with the change in the time span. Here you have on the left hand side you have the ages, and on the right hand side you have the changing diatom patterns. So likewise you can interpret the past changes. So the present day water quality database can also be interpreted using the statistical analysis. So you can do the PCA analysis. For example, we have correlated this pH with the Alopacida granulata. So likewise, you can use the PCA analysis to find out the relationship of the diatoms with the water quality. So I told you earlier that these dry periods are represented by the smaller soil size, whereas the wet period is represented by larger size diatoms because of the abundance of the nutrient level. So this lake, what we have studied in the central India, here we found the diatoms mostly found in the neutral pH. So that is very interesting. This is something called as more or less the boreal environment. So this is because of the abundance of the organic matter in particular lake. Likewise, various lakes will have the different history and different physicochemical parameters that can be understood by correlating the diatom assemblages and the present day water quality. So in summary, what I would like to say that diatom analysis is extremely useful in interpreting paleo-environmental conditions, but you need to follow the integrated multidisciplinary approach, and it must be used to analyze all of the available data. It could be the proxy one or the direct data. So considering the global crisis of the water, sedimentary diatoms can be used as the best tools in the years to come for the better understanding of sustainable environment. And even you can use these diatoms to study the monsoonal pattern, paleo-monsoonal conditions, and you may use that further to extrapolate it for the future trends of the monsoon, at least for the coming 50 years or so. So this is the literature. You can use the latest literature by the John, 2012. Now recently Karthik has published one handbook also. You can use that to find out the recent diatoms, how to identify these diatoms. Thank you very much.
Thank you, Dr. Sumit. We have got some questions. Fulmali, how one can determine sea level changes with the help of diatoms?
Hello. Can you
Hello?
repeat the question?
Are you able to hear me?
Yes, please speak.
Sure, sir. How one can determine sea level changes with the help of diatoms?
The voice is breaking actually.
Okay, sir. I will repeat once more, sir. How one can determine sea level changes with the help of diatoms?
So as I told you, the diatoms are very sensitive to environmental changes. Depending upon the climatic change, you will have the changes in the diatom speciation. For example, rise in the sea level. When the marine transgression is there, you have the abundance of the nutrient. So you can see the diversity of the diatom population. Diversity may be affected when you have the dominance when there is a regression in the sea level, you have the decline in the population of the diatom also. So first, there are several criteria that can be used, isotopic studies of the cell wall, and so many things we can use. But basically, you see the diversity of the diatom, planktic to benthic ratio. That is again very important to identify this.
So there is another question from on Mr. Sir.
Yes, continue.
Sir, there is another question from Mr. Irinjalakuda. Why does navicula usually occurs in group and in abundance?
No, navicula actually it is sensitive to the nutrient concentration. Wherever there is abundance of the nutrient, when the nutrient concentration is dominant there in any water body, you are going to get the navicular population. But it is not the case that in all the water body you are going to get the navicular, depending upon the availability of the nutrient and then the particular geology around the region and the water quality status of the particular region. So that decides the abundance of the particular diatom. It is not so that only navicula will be there. Along with that, the other forms you can get the centric diatoms as well. Ultimately,
So
whenever some organisms are dominating, it means there is the availability of the nutrient. Mostly the nitrogen, phosphorus, they are the common nutrients for the growth of the diatoms.
There is two more questions from Mr. Arun Joshi. The first one is, which are the biggest and smallest centric diatoms? And the second one is, how do...
Yes.
So I will read out the second question also. How do diatoms contribute in environmental changes?
Yes. So so far what we have studied in our study area, we could see the diatom as small as 2 microns, 2 micrometers. So it extends up to 100 meters, or in some cases it may be more than 100 micrometers, not meters, sorry, more than 100 micrometers also. Another question is how to identify.
How do diatoms contribute in environmental changes?
Yes, I told you, no, they are very sensitive to environmental changes, mostly the physico-chemical change, like pH, salinity. Even if there is a change in some pH, 0.1 pH, there will be change in the speciation. So the diatoms are used. So for example, your Alococera, I showed you in the slide, Alococera granulata is found in the eutrophic environment, wherein you have the abundance of nutrient. Whereas the tabellaria, what I told you, it is found in the oligotrophic environment. Likewise, the pH also, some diatoms are alkali biontic. I told you that one example is there, that is Pinularia. It is found in the more or less neutral pH, whereas some diatoms they are found in the alkaline pH. Likewise.
Okay, so there is one question from Miss Elora Priti Pushpa. What nutrients do diatom?
Yes, mostly I told you that they are the nitrogen and phosphorus, and the silica is also important. Nitrogen and phosphorus is important for the dominance of the diatom. These are the common nutrients. So these nutrients nowadays, because of the anthropogenic activity.
There is one more question. No problem, sir. Please continue, sir. Sorry,
because of the anthropogenic activities nowadays, through a lot of agricultural activities, excessive use of fertilizers, these nutrients are released through the soil erosion into the water body. So this impact is also need to be studied in the various regions of the country. This is very important to study how the anthropogenic activities are influencing the distribution of the diatoms. And to assess the status of the water quality as well.
Okay, so we have a question from Mr.
Yes.
I think I have some problem with my connectivity, sir. There is one more.
No, I could hear you. Yes. You can tell me.
So can you hear me, sir?
Yes, I can hear you.
Okay, sir. There is one more question from Mr. Brijesh Yadav. How can we use diatom for a quantitative fast sea surface temperature reconstruction from a marine core sample?
Basically, you need to adjust Dr. Rajesh Saraswat before the presentation. He explained very nicely. Once you collect the undisturbed samples, undisturbed sample collection is very important. When you collect the sample, the topmost portion should not be disturbed. No, undisturbed samples, when you collect, you can cut it into 2 equal halves and then study the internal structures and to decide the intervals. Topmost, generally in the leg sediments, what we do is we collect the topmost 10 centimeter sample. We cut the topmost 10 centimeter samples at 0.5 to 1 centimeter interval, whereas the bottommost sample may be extended up to 5 centimeter to 10 centimeter interval. likewise you have to decide, and then you should also see the annual bandings. So depending upon the different banding patterns, you have to decide the sample intervals, and then once you collect it, you can process. There is a specific procedure to retrieve only diadems to do the stable isotopes, oxygen isotope studies. Once you do the retrieve the pure diatoms, pure silica from the sediment, you can analyze it for the isotopic composition, oxygen isotopic composition, to interpret the paleo-C temperature.
Okay, sir. Hope the
But you
answer
need to
was,
see that the sample interval is very important from where you have to collect the samples. Okay, okay, continue.
okay, sir. Okay, sir. Sorry, sir. So there is one
Please repeat. Your voice
from
is cutting.
Parthasarathi Chowdhury.
Yes.
Sir, can diatom indicate marine transgression or regression?
Yes, yes, certainly. I told you, there is a rise in the sea level. You have the certain effect on the planktic benthic ratio. You can see the variation in the diversity of the diatom. When there is a transgression, the increase of the diatom. The regression, decline in the diversity. Likewise, planktic benthic ratio will also be affected. You can very easily identify by identifying these features. There is marine transgression regression. The region from the sediment, from the pores, marine pores.
Yeah.
Okay,
Hello.
sir. Now,
Yes,
hello.
yes, yes, sir.
Sir,
I
please
can
finish,
hear you.
Yeah. So
Sir,
sorry
I
to interrupt
think-
you.
sir, yes, sir.
Go ahead. Yeah. I mean, Subhangi also asked a similar question, and just now Dr. Humne has clarified the doubt. And Dr. Humne, we have listened to a very good lecture on diatoms and morphology, their applications, and with the case studies from the lakes of Maharashtra. And you have given a very good illustrative talk. And I'm happy to announce here that we are doing a collaborative research work with Nagpur University, Humne, and Samya Humne, and we have also published a couple of papers.
Right.
And
We have been studying the coastal lakes.
Yeah. And they have helped us in identifying the diatoms of a couple of our students. One of the student, Subhangi, she got her doctorate degree on diatoms, and another student is awaiting her viva, Sunita. So both have they helped in the identification of Dr. Yeah, Tarun.
Okay, thank you. Since late, so we are cut short. We are forced to cut short the session. So, with your permission, sir, Humne, sir,
Yes, yes.
I will
If there are
be,
no questions,
sharing your mail ID with the participants.
Yes, yes, certainly you can give my
There
email.
are some more questions, sir, but I think okay,
One
sir,
or
then
two-
I will
one or two
be
questions you can
sharing
take
your
up.
email ID with the participants.
You can share my email.
Then,
My email is here. Can you see my email on the first slide? Current
yes, sir.
slide.
Yes, sir.
It
Yes,
is
sir.
given here.
I'm
You
sure
can
that
share
the
this
participants
email.
also seen.
Yeah,
for the
and the
participants.
professor would like to say something. He I mean, he has to switch my.
I think
Okay.
we are.
So he is not switching microphone. Okay. Yeah.
Yes, I will switch on. Switch off the mic.
Okay, Tarun. Yeah.
Sir,
Yeah.
yes, sir.
Hello, Tarun.
Yes, sir. Yes, sir. You can speak, sir. We can hear you.
I want to stress here,
Yes.
and Dr. Hussain already knows it, that my students, Dr. Sumedh Humne, Samya Humne, Dr. Vanjarwadkar, Dr. Mude, Dr. Raut, they are doing excellent work on CA. So CA is not only CA is not only useful in oil exploration, CA also gives you job. It is clear, Tarun?
Yeah.
Hello?
You always appreciate your students and you encourage them, you know.
Yes. And my students are occupying my Dr. Raut is HOD and Dr. Humne. Soon he is going to become professor.
It's very clear, sir.
Yeah.
Okay. God bless you.
Okay. Tarun, go ahead.
Yes, sir. I think we can end this session. So I would like to invite Dr. Devi K for proposing the auto thanks to our speaker. Miss Devi, Miss.
Thank you, Tarun sir. I express my sincere thanks to Dr. Sumesh Humain on behalf of organizing team for his informative talk on diatoms applications in applied geosciences. Thank you, sir, for your talk.
Thank you, one and all.
Thank you, Humneswar. So I would like so to the participants, I think we are already late, but make sure you join the session by 2 p.m. So we are having two more talks by 2 p.m. The first talk is by Professor Devesh K. Sinha from Delhi University, and the second talk is by Dr. M. Suresh Gandhi from Madras University.
Before you conclude, Tarun, I would like to thank
Make
Professor.
sure you join by.
Dr. Humane is telling
Tarun, before you conclude, I would like to thank Professor Hussain
Yes, sir.
for giving me this opportunity to discuss my research work with the students to give the talk, today's talk. So I would like to thank Professor Hussain. I would like to thank Dr. Gandhi. I would like to also thank faculty members of the Christ College, especially Principal and Mr. Tarun. So thank you very much. Thank you.
Thank you, sir. The pleasure was always, sir. Having you in our speaker team was we were very delighted, sir. Thank you, sir.
Thank you.
So we can just break for the lunch.
And sharply by 2 o'clock, Dr. Devesh Sinha, he will come. Sharply by 2 o'clock, we will reassemble.
Sure, sir.
Yeah.
So let's dismiss for the lunch. Thank
Okay.
you for
Thank
watching
you.
and make sure you join by 2 p.m. Thank you.
2 p.m. Yes. Thank you.
मैं कहता नहीं मैं अरे हाँ वाला दिख रही चेन्नई वाला थोड़ा दिख रही अच्छा 1 बजे से उनका लंच हुई है ना देखा तला गया कल तक लाइव है एक जगह में है यूट्यूब में है बंद है लाइव चैट एक चार के आधार है नहीं कुछ नहीं आ रहा है ऐसे Usually the noise lecture.
Please check the sound. Am I audible?
Yes, sir. Yes, sir. You are audible, sir.
The voice is clear?
Yes, sir. It's clear. It's crystal clear, sir.